Malaria, a disease that has killed 1 million people and affecting another 500 million. However, scientists may now have a solution. Using genetic modification on the two major malaria vectors, they have made it so these mosquitoes express a gene that significantly decreases their vector competence. However, the expression of an exogenous gene can greatly lead to the reduction in fitness. So before chances of success can be judged and any release can be contemplated, a study named Impact of Genetic Manipulation on the Fitness of Anopheles stephensi mosquitoes was undertaken with the aim of understanding the relative fitness and population ecology of genetically modified mosquitoes. The null hypothesis of this experiment was that the frequency of the introduced gene should be subject only to genetic drift, whereas any fitness costs of the gene or process of transformation will lead to a reduction in frequency. With this null hypothesis, you would expect a Hardy-Weinberg equilibrium to be established after one generation. The four transformed mosquito lines were IV, VD12, ASML12, and MinRed. 200 larvae homozygous for the GM line and 200 wild-type larvae were chosen and then allowed to mate. Examination of their larvae under ultraviolet light allowed an unambiguous distinguishment between the wild-type larvae and the heterozygous and homozygous larvae for the fluorescent change genes according to intensity. The results of this experiment showed that in all cases, transgene frequencies were lower than expected in the first generation and then tended to decline over time, becoming extinct within 4 to 16 generations. Lines IV and VD12, which were of the same transposon construct, showed that the transposon had disrupted a coding sequence. Although this gene product appears not to be essential for the mosquitoes, it could be selectively disadvantaged. Line ASML12 dropped quickly in the first generation before remaining stable for 15 generations and then was completely lost. Line MinRed declined linearly after the first generation to extinction in the second. Scientists believe that the loss of the transgenic allele may be due to costs associated with their expression, their insertion in the mosquito chromosome, or through deleterious alleles that have become fixed during the breeding of homozygous lines. Overall results show that the genetic makeup of engineered lines has a major effect on the maintenance of transgenic alleles, and that in 3 or 4 cases the transgene itself or effect of its insertion has a fitness cost to the mosquito. They concluded that it would be more effective in the future if the mosquitoes were substantially outbred, that it can cautiously be concluded that the insertion and expression of engineered alleles will often reduce the mosquito fitness.